Okay, back with population genetics. You did a lab on this online, uh, but I haven't talked about it in class. So I wanted to at least give it a little brief attention here. Uh, as I ended off the last one, I said here's some definitions that you um, can look at. A definition: this is population. Uh, Population genetics is a combination of Darwin and Mendel's ideas about how populations change genetically over time. Uh, so again, more definitions here. What we mean by evolution, what we mean by gene pool, microevolution, and species. And I've got species in the red there because that's a definition I absolutely positively want you to know. Um, this is often on test 3, and I usually will put it as a short answer question, so you kind of have to be able to write out what a definition is. This semester, this material is going to be on test on the final, okay, not on test 3, but it's going to be on the final because it's after test 3. And I'm speaking to the spring 20 online class. So if I ever use this lecture again for anybody who's missed a lecture or something, that's who I'm talking to in this. Is the spring 2020 class. Uh, so anyway, on a normal semester, this is often a short answer question. Okay, all right. So it is highlighted, and I will probably come back to it. I mean, I won't come back to slide this slide, but I will keep referring back to that definition of a species, definition of a species. And I think I've already verbally told it to you in class a few times too, as well as we've seen it in a film. So it's something I definitely want you to know. I may figure out a way of having that on a multiple choice test. Okay, so still you need to be familiar with it, even though you may not need to regurgitate it like word for word and as a definition on a test. So the idea is that if there's no selection pressure on a population other than the pressures it's already adapted to, okay, once a population makes the change to a pressure and can exist under that pressure, then there's no need to change. So that's a non-evolving population at that point. It's reached an equilibrium with its environment. It's in balance with its environment. And so there's shuffling of alleles that accompany sexual reproduction. Your mitosis lab. Here we're talking about meiosis. Okay, had you look at how these alleles got reshuffled, okay, um, during the making of the egg and the sperm, the gametes. But that doesn't alter genetic makeup in the population, not really. I mean, it is the difference between eye color and hair color and that sort of thing, and that's not a change in structure or function or anything along those lines that actually like in human populations leading to evolution of any sort. Okay, that's just variation within the population. In Hardy-Weinberg equilibrium, the frequency of each allele in the gene pool will remain constant unless acted upon by other agents. So that's where your selection, your pressure comes in there. Unless there's a change, there's that equilibrium is going to stay distributed in a predictable fashion. So for that predictable fashion to maintain, we have these 5 assumptions that have to be met. And usually I have a lab in class. I told you we were going to do it right after spring break, and of course that didn't happen, but I refer to it in my lab as group sex. Reason is because we will you will mate with another student class. You got your little cards of alleles in front of you, and you're going to mix up your alleles and put them out there and make offspring. And you make 2, one for yourself and one for them, your partner to become. Then you become your own child, and then you have to go mate with somebody else because you don't want to inbreed. Okay, so we don't well inbreeding until the very end. There's a case 4 does do some inbreeding on purpose, so we can see what happens. But we do 5 generations. Okay, and so at that point in the class, if you didn't know everybody else in the class, you've met everybody else in the class pretty much because you've had to change alleles with them, and so that's where I give it that pet name, and it's not mine. I learned that at a science teacher education workshop one summer, and that's what the instructor called it. So I picked that up from him, not my own idea. So this Hardy-Weinberg equilibrium equation holds true if you're dealing with a very large population, which I always tell students in the classroom. Classroom is not a large population. On a good day for that lab, we usually get about maybe 16, 17, 18 students in there. And a large population is a few hundred, ideally. Okay, 500,000, even better. Remember I said that Mendel did his research with thousands of peas. I think maybe as many as 10,000 or more peas over the course of like 8 to 10 years. So population's large. Population's isolated. Okay, we don't have other species. Well, don't have other members of the same species with a different genetic sequence coming in. Okay. There's not any mutations that alter the gene pool. Mating is random, and all individuals are equal in their reproductive success. Now, these things in nature don't typically hold true. Okay, there's always some exception here going on. Okay. First of all, mating is hardly ever random. I tell the students they have to try to do that in the lab. They can't just go flirt with the cute guy or girl in the class by trying to mate with them, but they've got, they've got to uh, mate with everybody else in the class. There's one of the case 3, we do 10 generations. That one's challenging. When I tell you can't mate with the same person twice, you've got to go change alleles with somebody else each time, a different person each time. And so yeah, so anyway, random mating in the wild. Birds, everything, they have plumage, they have songs. Just picking on birds, other animals have their things. They're to attract the opposite sex. Okay. And the more attractive one is the one that's going to be more successful in breeding. So mating's not random. Okay. And therefore, again, that's the equal reproductive success too. So not all females will be able to birth live offspring, or even lay eggs that will all hatch with the same success as other females. So yeah, differences there. Okay, so, but we make those assumptions, and in laboratory conditions and things where we're studying populations or in enclosed refuges and things like that, we can somewhat ensure that some of those things are going to follow. For studying a small, or we don't want small, we want large, but still for study, sometimes we have to sacrifice one thing to get another. So we're, you can see what you can control. Okay, fruit flies in a laboratory. Now there you can get lots and lots of little things in a small space. You can study a large population. Okay, so that's why they're popular to study. So looking at the Hardy-Weinberg equilibrium equation, I think that lab online had you do it. I need to work through an example of that with you. So I may have to do a video where I just completely do that. I don't have an example problem in this presentation. Okay, didn't think about that ahead of time, should have. Let me see if I can come up with something real fast. And this will probably be, yeah, possibly pretty close to where we end this. We'll just go through a Hardy-Weinberg equilibrium equation and call it good. Okay. Whoa, get back here. This is where I want to be, I think. I want to go and insert a slide here if I can. So bear with me. All right. No, back. All right. I'm going to do this off the top of my head because I haven't thought about this. Oh, well, I guess I need to give a different color. That's the Hardy-Weinberg equilibrium equation. P squared plus 2pq plus q squared equals 1. You're like, oh my gosh, I didn't sign up for a math class. It's not at statistics. That's different. You've seen this equation. This is a really familiar equation to you, and you're going, no, I've never seen it. I swear I've never seen it. You're fibbing to me. Okay, well, let me rewrite it, because I do this, and I usually, in a classroom of students, I see the light bulb go on. They're like, oh, Okay, so let me just put it, and you saw it with other letters too. It's just X and Y get used so much in math class that this is what looks familiar to you. Though you saw it as probably A and B and M and N and S and T and whoops, I'm talking and I'm not paying attention to what I'm doing. That's a Y. Stay there. Okay. All right. Now I'm going to do something else. I'm going to come over here and I'm going to draw our friend the Punnett square. Except I'm drawing real sloppy, but that's okay. Usually I draw the sloppy on the board too, so this is just like being in class. All right, I'm going to have, I'm going to use A over A. Okay, or A, yeah, big A, little a. This is our dominant allele, and this is our recessive allele, okay? And I'm going to have two heterozygous parents. Come on, little a, there we go. All right. All right, so just like normal Punnett square, big A, little a, that's one parent. Next parent we'll bring around here, big A, little a. And now we're going to do our Punnett square, kind of like our little multiplication table, matching up type of thing, big A, little a, or big A here, big A, little a here. Big A, little a, and little a, little a. And it's like, okay, now how is the Punnett square related to this math thing she just threw up here on the board for us? This is ridiculous. Okay. For yet another killer. I don't know. Let's see. So, I think I'll go back to blue actually. Too many colors to choose from. I'm having too much fun with the palette. If we say that P is our dominant allele, and we say that Q is our recessive allele, two big A's would be A squared, right? Dominant A squared. I've got a big A and a little a, and I've got one and two of them. And then I've got two little a's here, so that'd be like little a squared. Yeah. So see how this equation represents our Punnett square? It equals the whole. Okay, something too that I don't think I mentioned in the actual genetics proper lecture is that this is not a representation of the children you're going to have. Exactly, put it that term. I like to use this as an example of this is dark hair and this is like light hair, red, blonde. Okay. Um, my husband and I both have dark hair, so this is us. And how do I know this is us? How do I know that we have the recessive allele? Because all you can see is our dominant allele. Okay. Well, it's because right here, and if you've seen my kids, you know I've got a redheaded daughter. Okay. I've had a redheaded son as well. And I've got a, well, my blonde is getting a little darker. That's a different gene. That's called a Teutonic gene trait of hair color. And I think they're still kind of studying what triggers a child to be born blonde and then get darker as they get older. But it's very common amongst the Northern Europeans. And that's why it's referred to, I think, as a Teutonic gene. Okay. The old Germanic tribe, the Teutons. So anyway, yeah, so we know we've got that recessive, each one of us, because we both had to have it to give in order to have redheaded kids. Okay, so but this, to point that out, doesn't mean that these two individuals will have three dark-headed children and one redheaded or light-headed child. Does not mean that. It means that every time my husband and I have a child, we have a 75% chance of having a dark-haired child and a 25% chance of having a red-headed child. And we've looked at each other and said, with our odds, we should be hitting the casinos because Yeah, 3 for 3. Yeah. So anyway, but yeah, the dark-headed, the blonde may actually be one of the dark-headed ones, but still two. Okay, two out of three. But that's it. It's a new game every time. It's a coin toss every time, with every child. That said, we can use this Punnett square to represent the expectation of a population. Okay, if you've got 1,000 individuals in a population and you've got a trait like this, dark hair, red hair, something, a simple dominant recessive trait, then you're not looking at possibility offspring of of two individuals. You're looking at what is the probability of finding those traits in the population. So now the whole population of like 1,000 becomes the one. Okay, that's the whole. One doesn't mean just one individual. One means the whole, whatever that whole is. It could be 500 individuals. We get 1,000 individuals. We get 638. Pick your number. It's the population you're looking at. You're studying. So here's where we look at the percentages. We say, well, 75% of the population should have the dominant trait, and 25% should have the recessive trait. Okay. And I didn't write my words out, but I want you to see that. Okay. So we can predict that in a population. Okay. We can look and see. So what I usually have the students do at this point, if I haven't already had you do it in a lab the previous week, is I have you do the taste papers with the PTC. I always have to be very careful how I say that. People say think I'm saying something else. PTC papers. This is a chemical that some people can taste and some people can't. If you taste it, you've got a dominant allele, at least one. And if you can't taste it, then you're recessive, so we know who you are. This is an interesting trait to study in the humans because it is looks like it's dying out of our population. At one point, they thought maybe it's an emerging trait, but they don't think so. But the dominant, we should have 75% of the population with the dominant trait and 25% with the recessive trait, and that's not what we see. We usually see it swapped around. Most of the students in the class are typically non-tasters, and a few, sometimes as few as one or none in the class are tasters actually. Okay, so and I am a taster, and I always apologize to my students. I know it doesn't taste pleasant. I usually let tasters go get a drink. Anybody can go get a drink at that point in time if they need to, but I will encourage that. I always have water with me. I always taste the paper ahead of time myself, just making sure that package of papers, if I especially open a new one, is working. But also, the students know that I'm not purposely torturing them without sharing in the misery myself. It's pretty puckery, okay? It's not really horrid. It's just pretty puckery and unpleasant, okay? So we use that one oftentimes to show that you can get an unexpected. But what that does is it allows us to determine about how many people in the population are heterozygous as opposed to homozygous dominant. Okay, I'm going to start with I'm going to give an example of something a little easier. This would have been a homework question problem in the lab that we would have done in class if we had class lab on this. Actually, I think I'm going to pause this here. See if I can pause recording. I don't know if I can do that or not. I can. All right. And be right